So, hello everybody, and thank you, Sarah and Anna, to have asking me to present today. I will talk about the French collection for plant-associated bacteria, which is called CIRM-CFBP. And I will also present a bit of the French context of MIRRI. So the collection involves a lot of people, and I will go through what we're doing right now. And I want also to explain how we moved from the single strains to complex microbiota. Okay, so first of all, the French MIRRI node. So I've put back the MIRRI map, so we're in France, and the node is, the coordinator is Jean-Luc Legras, one of my colleagues in Montpellier, and there is other people involved. So the French node, okay, yes, that, okay. The French node is constituted right now by two partners. The first one is INRAE. I'm part of this one. And the second one is the Pasteur Institute, and I will talk a bit more about that. Okay, so that's a map of the organization of the Pasteur Institute. So I will just go briefly. If you want to contact them, I can provide their contacts if necessary, if you have more questions. So they have central service for organization, and they are the project management office who takes care of projects, etc. And they also have in-house a platform for genomics. And then there are the collections. So the collection, which are called the CIP collection of Pasteur Institute, which regroups several different materials like bacteria, fungi, viruses, archaea, and phages. They have another collection which is slightly separated. It's the Pasteur cultures of cyanobacteria. It's historically they were not in the same organization, and actually it's also because the PCC, the cyanobacteria, there needs to be a growth continually actually, so they are not stored in the same way as the other resources. Then they have the National Collection of Culture of Microorganisms, which is the collection under the Budapest Treaty for patents, etc. So this one had to be separated. And then they have a collection for human health, and this one is more or less on the border of MIRRI and not always involved. And then you've got, can you go? Well, I can go through, oh, yeah. Thank you.
I have to be the one moving the slides. Sorry.
Thank you. And then there is INRAE. So INRAE is the French Institute for Agronomy, Food and Environment, and we are like 10,000 different people working there. And we are scattered all over the French territories, even abroad, even overseas. And a lot of different research institutes, research centers, I would say. And like a bit less than 20 years ago, INRAE decided to organize a bit more formally some of their microorganism collections. And that led to the constitution of the CIRM network, which is called CIRM for International Network for Microbial Resources, which is a big word to say that it's a network of 5 collections managed by INRAE. And all 5 collections inside, they are thematics. So you have the food-associated bacteria, which is located in Rennes with our colleagues there. You've got the the pathogenic bacteria, pathogenic towards animal or human, which is located in Tours. The yeast collection in Montpellier, the filamentous fungi in Marseille, and in Angers, my collection, which is the CIRM-CFBP for plant-associated bacteria. Can you go through, please, Sarah? And once more, please. So now I will talk more about the CIRM-CFBP collection itself and a bit context there. So we have resources which are strategic for plant health, actually. And because of the history of the collection and the team which included the collection, we have a really strong expertise in plant pathogenic bacteria. And I put two examples here. So bacteria attacking lamb lettuce or Xylella fastidiosa attacking olive trees in Italy. The collection, as I said, was created more than 50 years ago, and we now have more than 7,000 accessions, and you can find them through our website. I put the address there, and you can contact me if you have any question. The particularity of the collection is that we are embedded in a research team, which is called EMERSIS, and the team works on two main topics. The first one is the emergence and evolutive biology of plant pathogenic bacteria. So we're studying the diversity, epidemiology, how new diseases emerge and disperse over the place. And I have a photo of Xylella fastidiosa in Italy, and we had conducted a lot of works on this question, for instance, among others. The second axis of the team is around the seed transmission of microorganisms, of pathogens, of course, because there is pathogens transmitted by seeds, but we enlarge our topics toward the whole microbiota, actually, and how this microbiota on the seed can have an effect on the plant fitness later. Thank you, Sarah, for going through. As a CRB, we have missions. These ones are to preserve the resources and to make them available for the community, and of course to valorize that. And to answer to these missions, we have several activities, but mostly we went from the users' questions. And the first question often is, can you preserve this strain for me? So we have a deposit service, and to deposit strain in a collection can have many benefits, either personal for the researcher or also for the community. So I've got a short list there on the left, and that's in a publication we published in 2022, and which summarizes what you can do with that. So, and in blue on the map, you can see the places from which the strains originate today in our collection. So they come basically from all over the world. And we really thank all the contributors of the collection, because this collection can be really valuable for a lot of topics later. Then the second question is, okay, you have these strains now. To preserve the strains, how do we do that? We developed 3 different methods. We preserve them either in a -80 freezer or in liquid nitrogen tanks. So that's called preservation. Then liquid nitrogen is really stable over time and doesn't need electricity, even if you need electricity to produce liquid nitrogen. But the -80 freezer, which is really useful and really easy to use, you can have some evolution over the time, or you can lose your tubes because they can lost viability. We also, our main way to preserve the strains is to lyophilize them. It's all freeze dry. So you dehydrated them, and that takes a long time to us, and it's not, you cannot have that in every labs actually. So that's a real value, added value for the users, because it's one of the most effective way to preserve bacteria. So we have the facilities and know how to do that. What do we have in collection today? So I've talked about the number of strains, where they come from. We cover most of the major plant pathogenic bacteria, at least the ones attacking the aerial parts of the plants. And our specificity is to have not only one or two representatives of every pathogenic taxa, but to have a lot of diversity within every taxa, meaning that we have today more or less all the non-diversity of these plants' pathogenic bacteria, which can be really valuable. And because these bacteria can be pathogenic to plants, some of them are regulated. So we have some strains which are classified as group 2, that's for the human health risk, but these strains are minority in our collection. It's only for reference. We have some which are double use, a lot of quarantine organisms, and some GMO strains, and we are one of the few collections in the world having these GMO strains in the catalogue. So over the years, we also developed expertise in regulations, and I worked a lot about the Nagoya Protocol regulations and access and benefit sharing, because as you were able to see, the strains we have comes from all over the world, so that's had a real impact on our collection. So now the collection is registered at the European level among the collection compliant with the Nagoya Protocol, along with the SMZ. And after the questions to the need for users to preserve the strain, there is the need for distribution, because if you cannot use the strain, it's not real use actually after that. So, so in order to permit accessibility to the strains, we have a huge database keeping track of everything. We have an online catalog. You can find it on our website. And we have, of course, all the facilities, preservation expertise, and so on, and the expertise also in distribution. That's a benefit for this researcher, because when you publish something, you are supposed to give access to the resources permitting you to make the studies. But distributing a strain, particularly if it's under regulations, can be pretty tricky for people not used to do that. So the collections can do that. And today we distribute about 400 strains per year, and a third is for France, but the rest is for abroad, outside French borders, and about a quarter of them goes outside the European borders. And in order to improve our quality of service, the collection is ISO 901 certified for like 15 years now, and that works pretty well. Thank you, Sarah. So, and now what do we do with these strains? So still after this question of being able to preserve the strains and to distribute them, the users ask us, who is this strain? What is the identity? And that's not a trivial question, actually, because between the day the strains were deposited in the collection and now, a lot of things had happened, and the taxonomy of bacteria was completely revolutionized in the last decades, actually. So we had strains which were pretty well characterized when they were deposited in the collection, which had changed so many times of name, and the taxonomy has evolved, etc. We didn't know actually what they were anymore. So we decided to barcode all the collection. So we started by sequencing 2 housekeeping genes for that. And we're now moving towards complete genomes, because that's more and more accessible, and it's more precise, of course. So with that, we developed a strong expertise in plant pathogens diversity, but also taxonomy and identification. So we have phylogenetic trees for every strain of the collection by genera to be able to have a look to the diversity present there. We also made some interesting graphs about what the old names and the new names. You can see that on the left side of your screen. We also described several new species, and so on and so on. Can you go through, Sarah, please? And this huge database, because we have a barcode for each strain now, so we are pretty sure of their identification, permit us, for instance, to evaluate some identification methods. And like 5 years ago, we decided to evaluate the MALDI-TOF, because you may know that MALDI-TOF can be really useful to identify bacteria. And the databases at the time were mostly made for human pathogens and not for environmental bacteria and not for plant pathogenic bacteria. So we decided to build a database, and we tested because we know for all strains their exact identity. We tested against different strains in our collection, and that permitted us to determine that MALDI-TOF for the bacteria we studied can be really valuable to identify the genus, but may have some problems to go further away. That depends on the exact genera. And it was pretty important to make this analysis to be able to compare different identification techniques and to determine the limits of that. And then the question arising after that was, sorry, can you go back? Yeah. What the strains were able to do. So we acquired the biolog technology. Biolog permits to make the phenotyping, to test the metabolism of different strains, and you can compare them. And we have the Omnilog apparatus, which enable us to compare 50 to analyze 50 biolog plates in the same time, so to be able to compare a lot of strains at the same time, and that brought also a lot of data for that. And then the last question, which appeared in the last 5 years, was, and what about microbiota? So that led us to a new challenge. So can you go through, please? Yeah. So, well, BRCs are used to preserve some single strains, which is perfect. But what happens if we want to preserve a complex community? Can you go through? So that's a way to represent the life cycle for biological resource. First, you will sample it in the field, for instance. Then you will transport it, and you will include it in a BRC. There it will be preserved, stored for a long time. It will be amplified because we have cultivable collections, cultivable resources. And then it will be sent out once more to be used by the users. And some documentation may accompany that all along the process. And of course, there is a characterization also, which can happen all along the cycle. Okay, and if you want to apply that to microbiota, that's the next slide, please. And okay, if you want to go from single strain to complex community, that raises a lot of new questions. And when you sample a complex community in the field and you want to transport it, what about stabilization, and what will happen during this transport? And about the associated data, which ones are relevant? That may be not the same as the single strains. And of course, all the pre-analytical parameters. And if you want to preserve them, what the methods the best to do that, and what the impact of the storage. And if you want to send that out to be used elsewhere, once again, the transportation impacts. And once it arrives at the user's lab, is it still fit for purpose, and how you define it? So that poses also the question of the characterization. How do you characterize a complex community, and what the impact of the whole process there? And the documentation, we're not used to handle documentation for complex communities, so that maybe have some specific things, etc. Okay, so that's a lot of things. So can you go through, please? So we decided to team up with other partners in the micro project, which started 3 years ago, and it's still running for 1 year, and to start by going step by step. So can you go through, please? Because, of course, if you want to preserve communities, you have an heterogeneous community. And we know that within these communities, some members may be cultivatable, but not the others. So the preservation will have an impact, and the survival of the community may be heterogeneous, actually. And this heterogeneity in the survival may have an impact on the taxonomic composition. And then in turn, that may have an impact on the functional diversity. And I've looked at the literature a few years ago, and it's not better today. There is really a few studies about the preservation of microbiota, and very few of them, to say none of them, were dealing with the plant environment and the plant itself. And we also need to embrace the complexity and to accept that maybe there is some variation will be occurring there. So can you go through, please? So to go back to the micro project, because these questions actually are numerous, and the collection is run by only 3 people, so we cannot do everything. And we decided to go step by step. And within the micro project, we are partnered with 8 other partners, and we divided the work. And the objectives of the project is first to develop some technical innovations for sampling, preservation, or maybe targeted isolation, and also to develop the operational blueprints for microbiome biobanking. And as you can see on the figure, MIRRI is involved in this project, actually, because it's one of the infrastructure will benefit directly from that. So we have topics about preservation methods, keystone taxa, the data and the format and the preservation, the standards, the regulations. And we decided to focus on 4 different use cases. The human microbiome, that's just to preserve the nucleic acids, and the 3 other use cases to preserve the whole community as a living entity and not only the nucleic acids. Can you go the next slide, please? And among these use cases, there is the seed use case, and that's where I'm involved. So you can go through. So what did we do for that? So we decided to... Sorry? Sorry, there is an open mic somewhere.
You can continue, Perrine. We will mute it.
Okay. So we decided to share the experience among 3 partners in Austria, England, and in France. And the objective was to determine which procedures to minimize the preservation effects, but also to test if we were able to share experiments between different labs. And so we chose one preservation method, which was in liquid nitrogen. We worked from only 1 seed lot, the same seed lot, which was cultivated in France. And we tested 3 different cryoprotectants. We analyzed the microbiota at 4 different points before preservation, of course, and after 7 days, 3 months, or 12 months. We also tested the suspension, the coating actually in alginate encapsulation to making small beads of microbiota in alginate and or classical suspensions. And we also tried 2 different cooling conditions, either fast or slow, using a easy to use device. Can you go through, please? So what we did exactly is we extracted the microbiota from the seeds. We extracted that because we thought it was more relevant to have that out of the seeds. And then we made analysis before and after preservation, and the taxonomic composition was assessed by metabarcoding using 3 different markers. The metabolic profiling was done using biolog echo plates, which assess 31 carbon and nitrogen sources. And we also evaluated the cultivable fraction, because we may need to amplify the microbiota at some point. So we tested 2 different amplification methods, and we analyzed that also. And we decided to test 9 different conditions, with or without slow cooling, and the different cryoprotectants, etc. So you can go through. So the first results, we did not really count any colonies, but we took pictures of the plates to evaluate the cultivable fraction. And after culture, we sampled the growing microorganisms on the plates, and we extracted DNA, and we performed biolog also on that. So as we can see here, that was taken after 3 months of preservation in 2 different conditions, and we evaluated the bacteria and fungal fractions, cultivable fractions, and we can preserve some diversity. That was a relief, actually, because it works. Okay. However, as you can see, different conditions, different outcomes, and there is also variabilities between the different replicates of the experiments. So we need to go a bit further there, and we observed quite rapidly there that we had a problem with alginate conditions, because we were able to take out the bacteria out from the alginates, but the fungi did not really liked to be taken out, these alginate beads, and that can pose a problem later to microbiota manipulation. So it's only a technical problem there, but it can impair the experiments. So you can go through, please. And that's a quick word about the biolog results. So the eco plates are divided in 3 different replicates on the same plate, and there is some indicator, color indicator in the plate. At the beginning, everything's clear, and when the microorganisms or the community can make respiration, the control indicator will go from clear to purple, will be oxidized in formazine. So you can check quite easily which wells are positive and which wells are negatives. And here you have the summary of the 5 replicates assessed before preservation. And as you can see, there is some variability between replicates, but it gives an idea of what we expect of the community before preservation. Can you go through, please? And after preservation, here you can see a plot. This figure was to determine if there was differences between after preservation between the different time points at 7 days, 3 months, and 12 months, and it appeared that there is no real differences, no statistically significant differences between the time points. And also we found out that there were no significant differences between conditions. Okay, can you go through? But when we look better at different substrates, substrate by substrate, there is probably some significant impact before and after preservation. So the analyses are still in progress, so I won't say more about that today, but still we are looking at the fine effects of the preservation on that. Can you go through, please? And for the meta-barcoding, we now have the 16S, and it's still under analysis, so I won't be able to go really to conclusions today. However, we can see that it seems that there is no real impact between conditions either, but we need to go through to have a look to the finer modifications, etc. Can you go through, please? So the first insight, what I can see is that there is variations observed before and after preservation, but now there is no statistical significance. But we can say that the preservation of complex communities is possible. We still don't know exactly in which extent it doesn't affect too much the quality of that, etc. So the analysis is still in progress to understand the final scales of these results. And I just want you today to remember that, okay, preservation is possible, but we don't know yet how it's the real impact of that. And the perspective on this particular question is to obtain the last metabarcoding results and to analyze them, and of course to go through the biolog analysis and to integrate all the data together, and of course to integrate the microbe outcomes in our processes in the collection. Can you go through, please? And okay, I just want to conclude saying that biological resource centers are services for the community, and our collection, like a lot of the collection, evolved over time with the needs of the users. So can you go through? Yes. And I've listed 2 challenges we're facing now. So the first one is about scaling up. I talked a lot about microbiota, but actually we also have other things in the scaling up department going on, so that it could be good to have all the genomes of the world collection, but it can take a lot of time. For instance, with the development of microbiota studies and ecological studies, we'll need to devise high throughput preservation processes, which will be radically different than what we do now. And there is the other challenge is about more structuration, because I've talked about MIRRI, but at the INRAE level, we have this network of 5 collections, but we also have another network at the French level, and each different structure has its own exigence, on demands from us. So that's another challenge, actually, to try to navigate all these different scales and different levels of interactions between us. So that these challenges for the threats to the collections, and the bigger one is the HR, human resources, because we cannot do all of it, of course. But there is also a lot of new opportunities from that, because we have the opportunity to develop new expertise, to go toward new users, and to go toward new projects and new collaborations, and to divide the work, actually, because being in structured gives us more visibility, open us to more projects, and increase the sharing between collections, and to share a bit of the burden. So that's okay, it's challenging, but it can be really good in the end. Can you go through, please? And that's it for today. Thank you for your attention.